Hi everyone. Today I'll be showing how to prepare a DNA library using the RepliG kit. So the RepliG kit, unlike the Amplify DNA whole genome amplification kit, is meant to amplify a whole genome from either the cDNA or your DNA. So, and it's designed so that both reactions or both experiments can be performed simultaneously. So, so here my starting material would be DNA extracted from wastewater sample. First, I'll be making two aliquots. The first aliquot is for the whole genome amplification from cDNA, while the second one is for the whole genome amplification from DNA. First of all, I'll start by cleaning my workspace because the RepliG polymerase or amplification kit is very sensitive. So I have to first start by cleaning my workspace and workbench with 70% alcohol. So the next thing I'm going to do is to clean my pipettes. So in this ice box, I have all my samples, 10 samples, and all consumables for this exercise. So I would be making aliquots, 2 different aliquots for each sample. So that's the beautiful thing about RepliG kit, is it's faster than Amplify, because in Amplify to synthesize cDNA, I'll be using a different kit if I want to amplify the whole genome using the complementary DNA as my starting material for the Amplify whole genome amplification kit. But for the RepliG, I can use directly the NA extracted from wastewater. So for my sample 1, I'll take 2 microliter. So I'll do the same for sample 2. Sample 2, I'll take 2 microliter each for aliquots meant for the cDNA synthesis and whole genome amplification using DNA. So I'll take another aliquot from sample 2, 2 microliters. So I'll do the same for sample 3. I'll take 2 microliters twice. I'll do the same for my 4th sample. So for my control sample, I'll add 2 microliter of nucleus-free water. 2 microliter of nucleus-free water twice. So I'll add for the tube meant for whole genome amplification using cDNA, and for the tube meant for whole genome amplification using the DNA. The reason for both is for efficient, for efficient comparison between both materials as well. So next thing I'm going to do is I'm going to add genomic wipeout buffer to the tubes meant for cDNA synthesis. So the genomic wipeout buffer, what it does is it cleans out the DNA and leaves RNA in our tube. So I'm going to add 2 microliter of genomic wipeout buffer to the tubes meant for cDNA synthesis. Lastly, I'm going to add genomic wipeout buffer to my control. So after the addition of the genomic wipeout buffer, next thing I'm going to do is to add 8 microliter of water to bring the total volume to 12. So I'm going to add 8 microliter of nucleus-free water to all tubes, to the tubes for the DNA and to the tubes for the whole genome amplification using the cDNA. So for the tubes for the whole genome amplification using the cDNA, I'll have a total volume of 12. For the tubes for whole genome amplification using the DNA, I'll have a total volume of 10. So lastly, to my non-template control, I'm going to add 8 microliter of nucleus-free water as well. So I'm going to centrifuge. So for the tubes for the cDNA, I'm going to set up a PCR run. This is because in this tube I added the genomic wipeout, the genomic DNA wipeout buffer. So I have to incubate at 42 degrees Celsius for 10 minutes. So while this is going on, I'll prepare my whole genome amplification ready mix. So the whole genome amplification ready mix is for the it's a mix for the genomic DNA. So in this mix, I'll be adding to the other tubes where I have the just genomic DNA. So I'll also use the genomic wipeout buffer in this case, but in this reaction, the actions of the genomic wipeout buffer is more like limited, but the genomic wipeout buffer has some elements that aid this reaction as well. So I'm going to prepare the mix. So I'm going to add 46 microliter of my reaction buffer. After that, I'll add 23 microliter of the genomic wipeout buffer. And after that, I'll add 11.5 microliter of nucleus-free water. So always mix by pipetting 2 to 3 times. Then add 11.5 microliter of my endonucleus, of my exo-resistant primer, sorry. Then I'll add 11.5 microliter of my exo-resistant oligo-DT primer. And next I'm going to add 11.5 microliter of my whole genome amplification enzyme. Next thing I'm going to do while my reaction is still in the PCR machine is I'm going to prepare another mix for the cDNA that I'm going to synthesize using the RepliG kit. So I'm going to prepare the quantitative script reverse transcriptase mix. Always remember to label your tube properly. I'm going to take 46 microliter of my reverse transcriptase polymerase buffer. And 11.5 microliter of nucleus-free water. 11.5 microliter of my exo-resistance random primer and with DT primer. Then 11.5 microliter of my quantitative script enzyme reverse transcriptase. So I'm going to mix by pipetting 2 to 3 times. So when I'm done with both mixes, I'm going to flick, then centrifuge. So next thing I'm going to do is I'm going to take my reaction from the automocycle. I'm going to centrifuge. So the tube meant for my cDNA for amplification of genome amplification using the cDNA. I'm going to add 8 microliter of my quantitative script reverse transcriptase enzyme, so which is the tubes in this second line. I'm going to add 8 microliter. So what I'm going to do is I'm going to open all tubes because I want to save my tips. So since I'm adding same volume of enzyme mix, I'll add without making contact with the rims of the tube. So for the tubes meant for whole genome amplification using the DNA, I'm going to add 10 microliter of my whole genome amplification mix. So I'm going to add 10 microliter. So when I'm done, next thing I'm going to do is I'm going to centrifuge all tubes for both reactions. So after that, I'm going to incubate both reactions at 42 degrees Celsius for an hour and 95 degrees Celsius for 3 minutes. So when the reactions are in the thermocycler, next thing I'm going to do is to prepare a ligase mix. So I'm going to prepare a master mix. So I'm going to add 92 microliter of my ligase buffer. And I'm going to add 23 microliter of my ligase enzyme. Now I'm going to mix by pipetting 2 to 3 times. I'm going to centrifuge. So when the reaction and the thermocycler is done, I'm going to take it, then centrifuge. So I'm going to add 10 microliter of my ligase mix to all tubes. So using the P20, I'm going to add 10 microliter. So I'm going to mix by pipetting 2 to 3 times. So my both control samples, I'm going to add also 10 microliter of my ligase mix. So next thing I'm going to do is I'm going to centrifuge again. So when I'm done, I'm going to incubate this reaction at 24 degrees Celsius for 30 minutes and 95 degrees Celsius for 5 minutes. So at 24 for 30 minutes and 95 for 5 minutes. So when the reaction is in the thermocycler, next thing I'm going to do is I'm going to prepare another mix. So I'm going to prepare another master mix for the RepliG enzyme mix. Remember, always note and remember it's nice and better to always prepare the ligase master mix and the RepliG master mix 5 to 10 minutes before your PCR machine, your reaction in the PCR machine is done, and always remember and note to prepare them freshly each time you want to perform this reaction. I'm going to add 300 microliter of my RepliG buffer. So I'm going to add 300 microliter of my RepliG reaction buffer. Then next, I'm going to add 11.5 microliter of my RepliG enzyme. I'm going to mix by pipetting 2 to 3 times. And I'm going to centrifuge. So after 5 minutes, I can take my reaction from the PCR. I'm going to spin down. So next thing I'm going to do is I'm going to add 30 microliter of my RepliG mix to each tube. So to all tube, I'm going to add 30 microliter. I'm going to mix by pipetting 2 to 3 times. So when I'm done, next thing I'm going to do is I'm going to set up the last PCR reaction, which is 30 minutes for 2 hours and 65 Celsius, which is 30 degrees Celsius for 2 hours and 65 degrees Celsius for 5 minutes. 35 for 2 hours and 65 for 5 minutes. So when the reaction is done, this I can store in -80. Afterwards, carry out an endonuclease treatment, which I've shown in the previous video. So after the endonuclease treatment, I can go ahead with preparing the DNA library for Oxford nanopore sequencing. Thank you.